Good morning. I am D. Parthibane, a tutor in the Department of Biochemistry, Lakshmi Narayana Institute of Medical Science. Today, today topic of title is recombinant DNA technology. The development of recombinant DNA has revolutionized biology, having an increased impact on clinical medicine, manipulation of a DNA sequence, and the construction of chimeric molecule called genetic engineering, novel molecular genetic tools to examine the genomic sequence at the molecular level. A DNA is a very big structure, and how there are molecular scissors are available. These scissors are able to cut the DNA. The cut fragmented DNA can be cloned into a foreign vectors. The molecular scissors are generally a restriction enzyme. These restriction enzymes are very unique and sequence specific. And these enzymes are generally present in the bacteria. The few of the listed endonucleases are the EcoR1, BAMH1, and HIN3. These are all the enzymes which cut the DNA to produce a sticky patch. However, the other type of endonucleases, which is a H-pal, which cut the DNA to release the blunt end. These enzymes play vital role, especially in the molecular cloning and recombining process. We will discuss some of the properties of these restriction enzymes. Endonuclease is an enzyme that has the ability to cut the DNA at a specific DNA sequence within the DNA strand. It is a key tool in recombinant DNA research. The endonucleases are called as a restriction enzyme. Why it called a restriction enzyme? Because it restricts the growth of the bacteriophage. The bacteriophages which infect the bacterium and causing death of the bacterium. This is one of the defense mechanisms that safeguard the bacterium. The restriction enzyme have a specific sites of that. These respective specific sites will not cut the own DNA. This own DNA is prevented from the restriction endonucleases by the presence of another enzyme called methylases. These methylases specifically methylate the sequence specific manner, thereby inhibiting the enzyme to access into the own DNA. Thereby, these enzymes have the ability to prevent the genomic DNA of its own. The DNA, there are two different of cuts made by the endonucleases. One is a blunt end, another one is a sticky end. The blunt end and sticky end is also useful, responsible for making a recombining process of a DNA. The sticky end of the vector or any fragmented DNA which reconnect to themselves and anneal to form a tandem heterogeneous repeat. This would be a contiguous manner of a gene. This must be prevented for the molecular cloning. How to prevent it? How to circumvent this problem? Another enzyme which is added to the respective N terminus called terminal transferases, it add a nucleotide at the 3' end. Thereby we can make a sticky patch. This process is useful for the unhealing and it create a homopolymer training. This homopolymer tailing is an important technique, especially the ligating two different ends of the DNA. How to make a chimeric DNA clone? Here, we use a plasmid. A plasmid is an extra chromosomal apparatus which is present in the bacterium. These extra chromosomal apparatus have a distinct functional role. By exploiting this plasmid, we were able to clone our foreign DNA of interest. The purified the plasmid is then added with the restriction enzyme. These restriction enzyme having a specific a site sorter where the specific site is cut open, and the same similar enzyme is added to the DNA of interest, which is want to be cloned. That is the foreign DNA. Example here, foreign DNA, our interest is mainly to clone the human DNA. And the same enzyme is access to the genomic DNA of a human and get the fragment, and that fragment which is having a similar sticky end on the either side of the not only at the foreign DNA but also at the vector region, and add these two together, the plasmid as well as the foreign DNA. Due to the presence of a sticky end, it goes and bind and transiently then add the enzyme called a ligase. These ligase which does the phosphodiester bond connection between the vector as well as the foreign DNA to form a complete the vector molecule which is loaded with the foreign DNA. This technique is called as a recombinant DNA technology. By using this technology, there are various other enzymes are used in the recombinant DNA technology. Those enzymes are alkaline phosphatase. The alkaline phosphatase, the biochemical action is dephosphorylation of 5' end of the either RNA or the DNA molecule. BAL31 nucleases. It degrades both ends of 5' as well as 3' end of a DNA. DNA ligase. It catalyzes a 2 DNA molecule by providing a phosphodiester bond. The DNA polymerase 1. It synthesizes a double stranded DNA from a single stranded DNA. The DNA is 1. This enzyme under the appropriate condition produces a single stranded nick in the DNA. Exonuclease 3. These exonuclease 3 remove the nucleotide from 3' end of the DNA. The lambda exonucleases. These lambda exonucleases remove nucleotide from 5' end of the DNA. The polynucleotide kinase. This enzyme transfer the gamma position of ATP into phosphorylate into the 5' end of the OH group of either DNA or RNA. A reverse transcriptase is an enzyme which is synthesizes the DNA from RNA template. SN1 nuclease is degrade the single stranded DNA. The terminal transferase which can add a nucleotide to the 3' end of the DNA. What are all the classical properties of the vector? This vector is the carrier molecule which can carry not only the extra chromosomal apparatus of a bacterium, but also there are some large dead spaces are there. These dead spaces we can cut open and insert our gene of interest or the sequence want to be cloned. This is the property of, by using this property of the vector, in the recombinant technology, by exploiting the recombinant technology, we can design our own vectors, which are all the prime sites, which is important for the replication. Those prime sites and other important sites, if we unloaded into a vector, thereby this vector can be used for the cloning of a different gene or a stock, the genomic stock can be compiled. What could be the properties of the vector? Generally, the vector property, a drug resistant gene must be present. The name of the vector is TB322 is a vector which is responsible only for the cloning, And PR set vectors are different types of vectors are there. Those vectors also responsible for making a cloning as well as the protein expression in the bacterium. And there are multiple cloning sites where the gene can be cloned by you providing a different restriction enzyme can access into it. Here in this picture, you can clearly see a fragment of DNA which is cut open and then inserted in the in this region. Thereby, you are cutting and inserting one of the drug become resistant to that. By the screening method, you can identify the vector which is encoded with the DNA. And to do the vector to do the cloning, different fragment length cloning can be carried out. The vectors is depending on the DNA size, which ranges from 10 kilobase per to the 3000 kilobase per. Is all the insert of the huge molecule is all depending on the properties of vector. Here I have listed five properties, five different types of vector. The first one is the plasmid ER3Q2. This vector can take up to 10 kilobase per fragment. The second one is a lambda fetch, which can take up to 10 to 20 kilobase per. The next come is in a cosmid vector. It can take up to 35 to 50 kilobase per. The next one is the bacterial artificial chromosome, which can take up a fragment of 50 to 250 kilobase pairs. Then the yeast artificial chromosome. This is yeast artificial chromosome will take a 500 to 3000 kilobase pair for the cloning. The properties of the plasmid vector. The plasmid vector must contain an origin of replication, which is required to propagate the plasmid and maintain its copy number at 10 to 20 copies per cell. And it must have two antibiotic resistant gene. One is the tetracycline and ampicillin resistant. In order to better identification of the cloned fragment is present inside the DNA, inside the plasmid. And the plasmid must have a multiple cloning site where having a different restriction enzyme can access into it. And it also facilitate the plasmid also small enough to facilitate entry into a transformed cell. And once it get transformed inside the cell and make the protein for the transgenic protein to get expressed and easy for the purification of those proteins. This is the property of the plasmid vector. It must have a shuttling vector, either the vector to provide a cloning as well as the expression of the protein of interest. Now, once the cloning is done, this is a recombinant plasmid is done. This recombinant plasmid is then transformed into a E. coli cell in the presence of a calcium chloride method. The calcium chloride which makes a pores. In that pore, it extends at the 42 degree of heat shocking for 1 minute time. The pores will be enlarged along with that calcium chloride. The DNA also passes into the pore, get entry access into the bacteria, where it is a transformed. These transformed bacteria, non-transformed one, is plated in a ampicillin coated plate. Wherever coated in a drug plate, the drug, wherever the plasmid is there, you will find a colony. These colonies having the recombinant plasmid is present, and you can purify the plasmid based on the plasmid purification technique. And then want to go for expression, you can go for the expression, or you want to keep it as a stock for the further investigation or gene protein interaction, everything. It can be studied by using this vector unloaded DNA fragment. There are other molecular biology techniques are there. Those techniques are very useful in the study of recombinant DNA technology. The first technique that we are going to look at is the polymerizing chain reaction. It is called as a PCR. These PCR having a three unique different steps. The first step is a denaturation. The denaturation of the given DNA at 90 degree. And second step is the annealing of the DNA and its primer, respective primer at the 55 degree, and polymerization start at the 75 degree. All this process, it requires the polymerization is mainly done by the heat stable DNA polymerase, which is isolated from a thermus aquatic gas. These DNA heat stable polymerase can withstand up to 95 degree temperature. With this heat also, this enzyme is not have the ability to denature. It does the polymerizing function. The PCR, the DNA fragment we took must be amplified for that the forward sequence as well as the reverse sequence. The forward sequence, forward primer is added, and the reverse sequence to be synthesized, reverse primer is then added. Under the process of denaturation, all will be remain as a separated. When the annealing happens at the 55 degree, the forward and the reverse primer will bind to the DNA of interest, and then the polymerization process start at the 75 degree. You can see here the cycle one, the DNA get polymerized to have two bands, and then the cycle will keep on continuing with all these three processes. Thereby, there is increase in the PCR will go run for 35 cycles. Once it runs for these three even for 35 cycles, the PCR, the respective DNA is expanded, amplified. What is the application of PCR in the medicine? It is applied in the field of to detect the infectious agent, especially the latent virus period, and it is useful in making a prenatal genetic diagnosis. And it is also useful in detection of allelic polymorphism of a gene, and it also established in the precise tissue a transplant one between the xenografting between the foreign and the host reaction. And it also useful in studying the transcript libraries for a given tissue using RT-PCR method. And it is also useful in construction of studying the genomic sequence, genomic library for a given tissue. The second technique is the sudden blotting technique. The sudden blotting technique is used to confirm the presence of the foreign DNA fragments. First, isolate the three sudden blotting technique is generally used to confirm the foreign DNA fragment, especially the transgenic animal preparation. Here you can see the DNA molecule. The DNA molecule, once it is purified from a given cell and then cleaved by the restriction enzyme, you can see here the fragments. These after restricted enzyme is then subjected to the agarose gel. You can see after electrophoresing, you can see here the fragments. These fragmented gel fragment is then blotted onto PVDF membrane. Blotting technique. After blotting, the DNA is then loaded onto the PVDF membrane, and this PVDF membrane is used to hybridize with the various the respective DNA or RNA probe having a radioactive material at the at the phosphate is radio labeled, and then you can detect the DNA of interest. And this is the one way to confirm the presence of foreign DNA. This technique is called as a sudden blotting. Then the protein expression and the expression of mRNA is then confirmed. The confirmation of the mRNA is conferred by the Northern blot technique. The same in a given tissue, in a given cell induced with the drug and another one not induced with the drug. We can see and isolate the RNA and then resolve the RNA in a RNA gel, that is the agarose gel. Then the RNAs are separated again, it blotted transfer into the PVDF membrane. Either you can use PVDF and these segregated the fragment is then probed it with the DNA probe. Either it may be a chemiluminescence or the radioactive material. With this radio autography, you can able to detect your RNA of interest. This technique is Northern blotting. Northern blotting generally used to confirm the expression of a mRNA in a given tissue under any influence. And it is also useful in studying various oncogene expression also. Then comes the Western blot technique. Here the protein, the whether the protein is expressed or not. To confirm the protein is expressed or not, first isolate the protein and then resolve the protein in a phage electrophoresis. After resolving it, you can see here a lot of different bands where this separation is mainly based on molecular weight separation and is then transferred into a PVDF or nitrocellulose membrane. Once a protein all blotted into the membrane by using a wet to transfer technique, then the 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 PVDF membrane is then blocked with the 5% blocking solution. After blocking with the blocking solution is then probed it with the primary antibody followed by the secondary antibody having a coloring reagent. This coloring reagent will give a signal. Here you can see a black color signal of a respective protein. And depending on the quantitative expression of the signal, we can signify that these proteins are expressed in a higher or the lower magnitude. Now, the DNA was fragment is then cloned it into the vector. And we don't know what exactly the sequence of the DNA. To study the DNA sequence, we use a DNA sequencing technology, that is a Sanger's enzymatic technology to study the sequence. And here we did a dideoxynucleotide, which is a dideoxynucleotide which goes and bind to the terminus end. This dideoxynucleotide, which is having a fluorescent material, and this fluorescent material, it goes and bind, no longer bind the other molecule next to it, appending to it. Thereby the terminal stop, a terminal stop at the DNA biosynthesis. You can see here in this picture, here they added a different nucleotide, different version of the nucleotide. There is the sequence getting synthesized at the terminal position, it gets stopped. The star one, which indicate that either it may be chemiluminescence or the radioactive material. And all the synthesis in the 4 different tubes. A single fragment is added to the 4 different tubes and add all other nucleotide along with that, the D-deoxynucleotide also. The fragment are allowed to synthesize by adding a DNA polymerase into it. You can see a different fragment length and then load it onto a polyacrylamide gel. You can see here a different fraction is by naming one fraction, you can able to deduce the sequence of a DNA. This method is called as a Sanger's method. There are other method is also available, that is a chemical method. Maxim and Gilbert method also available. But the Sanger's method is a classical method used for the DNA sequencing technique. Then comes the preparation of a transgenic mouse. How transgenic mouse is prepared? They took whatever the DNA you want to insert, a foreign DNA into a a ovum or ovum, you have to collect the fertilized egg and then insert your cloned DNA into the pronucleus. After the cloned DNA into the pronucleus, where this DNA having similar to that site where it get exchanged, and after exchanging, your DNA of interest is then inserted into the genomic DNA of the given egg. Once you get then the worm is then implanted into a pseudo-pregnant female and allow the female to produce a offspring. When the offsprings are produced, these offspring, whatever the litters that it form, do the genomic DNA confirmation, especially the southern blot technique, to identify the presence of the foreign DNA is present in the pub. Once the genomic DNA was confirmation, generally, generally about 10 to 30% of the offspring will contain a transgenic one. And once the transgene was confirmed, is then used for the second next line of breeding. Thereby, you are getting a transgenic mouse. In the transgenic mouse, you can study the protein and their function. This is the pinnacle of the recombinant DNA technology. Then the human genome library preparation. For that human genome, it contains a very vast, so many kilo base pairs are there. How to make a library from the vast DNA preparation. This is a human DNA. This human DNA is partially cut by various restriction endonucleases, which is a fragment length of 20 kilo base pair having a sticky ends. The same bacteriophage DNA, which is having a 49 kilo base pair, is then used for loading the foreign DNA. The same enzyme must be cut and then remove the portion of the DNA which is not required for any function of bacteriophage, then replacing it with a 20 kilobase per fragment and anneal and then ligate the fragment. Then this DNA is called as a recombinant phage DNA. Once recombinant phage DNA is made, is then packed into a bacteriophage. Likewise, we can able to make different libraries. Whatever the essential gene which is present in the human, all the genes and other portion of the repetitive sequence, everything is can be loaded into a recombinant into a bacteriophage. And these bacteriophage, you can set up a library whenever any gene of interest is undergo mutation or anything. From the library, we can pull down and then do the protein expression and do the transgenic study in order for the betterment of the disability. The last one is restriction fragment length polymorphism. This is the fragment of the DNA which is isolated from the human being, is then subjected to any restriction in endonucleases. When the endonucleases get access into it, we get a different fragment length, and this different length which is in polymorphic manner, that's why it is called as a restriction fragment length polymorphism. Here, one classical example to study the sickle cell anemia 1. Here it is a beta globulin gene, and the beta globulin gene in the normal phase, it will do the PCR fragmentation, PCR with the primary and forward and reverse primer. You can see here a band of 1.15 kilobase per 1.15 kilobase are up to here. Since there is a mutation in the sickle cell, this mutation which will lead to change in the base pair, and you will have a 1.3 kilobase per DNA. And the pedigree analysis of the two heterozygous made well father and mother heterozygous for the sickle cell anemia, and you can see the letters with the homozygous one. The homozygous one is a disease will be pronounced. And this is a classical example that we can study the RFLP technique. Now, the recombinant DNA technology is used in various molecular analysis of the disease. The first one is a normal variation of the gene. Any gene is normally undergo some kind of polymorphism, especially in the intonic region. And the gene variation which causes a disease, that is a classical example of any single or a a mutation which can cause a disease, the sickle cell anemia, etc. And the point mutation. Point mutation is the cystic fibrosis, sickle cell anemia. There is a gene, one nucleotide change in the one nucleotide, which causing the, the mutant. And the pedigree analysis. The pedigree analysis generally will be carried out by using a restriction fragment length polymorphism. Then the prenatal diagnosis of the kids. We use a PCR technique, RT-PCR, to study the gene mutation and whatever the disease that the baby is going to suffer, and then able to easily able to detect it. Then the microsatellite DNA polymorphism. This microsatellite DNA polymorphism is very important one. There is a satellite, the repetitive sequence which is remain in a very repeated manner. For example, GC is a repeater which is contiguous for 100 nucleotide. Then these type of repeats are called as a microsatellitism. This polymorphism is unique for a given person. And based on this microsatellite polymorphism, we can able to detect one person varying to the another one. The final one is the gene therapy. In the gene therapy, in the gene therapy, the foreign gene, for example, the cystic fibrosis. Cystic fibrosis, the gene which is a point mutation, which is responsible for causing this one. If you nucleotide addition by putting back a proper nucleotide into the gene, we can modify and the disabled gene become a normal gene. This technique is called as a gene therapy, and the gene therapy now currently is applicable in the lot of various clinical trials is also undergoing. Then the preparation of the transgenic animal. These transgenic animal are also useful in the studying the recombinant DNA technology uh, for the expression of the protein as well as any oncogenic expression, etc. Different types of genes, and we can characterize their function. Then the RFLP, VNTR. These are all the different techniques which is useful in studying the recombinant DNA technology. With this, I am concluding my talk. Thank you very much for listening. Stop.